Good afternoon, students. Now, students, today we are going to discuss with the topic meiosis of the cell division. Okay, we have already discussed about the mitosis topic of the cell division. Now, today we are going to discuss with the meiosis of the cell division. Now, let me tell you meiosis, what the meiosis is and what are the changes which occurs during the process of the meiosis. Okay. So first of all, meiosis, the term meiosis, it was proposed by the name of the scientists called the Farmer and Murray. So these two scientists have proposed the term meiosis. Now, what meiosis is? Meiosis is a type of the cell division which is going to be useful to convert the diploid cells into the haploid cells. Okay, so this is a cell division where in this cell division the diploid cells, they led the formation of the haploid cells. Okay, and in that process the whole meiosis which occurs in special type of the cells. The meiosis is a cell division like you have gone through in the mitosis first. Mitosis occurs in only somatic cells. Okay, so in the similar manner, meiosis that occurs, it's a special kind of the division which occurs in a special type of the cells only. Okay, and that special sized of kind of the cell division are responsible to reduces the chromosome number by the half. So which reduces the chromosome number of this individual to the half in the production of the haploid cells. It means through the process of the meiosis, the diploid cells get converted into the haploid cells. Okay, so this kind of a division is known as meiosis. It is known as meiosis. Let me tell you, meiosis occurs during the life cycle. If you check, like I have told you in mitosis, कि mitosis जो बेटा होता है वो special somatic cells में होता है, body cells में होता है, but meiosis जो है ये special cells में होता है. Those specialized cells are called the germinal cells. They are known as germinal cells. So meiosis जो है, meiosis occurs, it occurs in germinal cells. It occurs in germinal cells. So beta, so germinal cells में ही meiosis होती है, सारी cells में नहीं होती. First of all, so germinal cell becomes the specialized. They are the specialized cells. Second thing, remember, meiosis occurs during the life cycle of the individual. कोई भी इंडिविजुअल, कोई भी ऑर्गेनिज्म जितना उसका लाइफ साइकिल होता है, जैसे आपने माइटोसिस में देखा कि नंबर ऑफ सेल डिवीजन जो है माइटोसिस में वो कंटिन्यूअस प्रोसेस होता है थ्रू आउट द लाइफ, बट मियोसिस थ्रू आउट द लाइफ नहीं होता है. किसी भी ऑर्गेनिज्म में मियोसिस जो है वो एक पर्टिकुलर स्टेज पे होता है, ड्यूरिंग द लाइफ साइकिल पर्टिकुलर स्टेज पे ही होता है, वो थ्रू आउट द लाइफ नहीं होता है. सो रिमेंबर, meiosis ensures the production of haploid phase in the life cycle of the sexually reproducing organisms where the fertilization restores the diploid phase. ठीक है, so meiosis occurs during the gametogenesis, यानी जब एक ऑर्गेनिज्म, डिप्लॉयड ऑर्गेनिज्म जो होता है उसमें जब गैमीट बनते हैं तो वो गैमीट बनते हैं meiosis से. जब उसमें meiotic डिवीजन होता है तो डिप्लॉयड सेल से हैप्लॉयड गैमीट्स बन जाते हैं. ओके, सो meiosis occurs during the gametogenesis that leads to the formation of the haploid gametes. This is the main function of the meiosis. कि meiosis जो है वो diploid cells में डिवीजन करता है, न्यूक्लियर डिवीजन होता है, फिर साइटोकाइनेसिस हो जाती है और उससे डिप्लॉयड सेल जो है वो कन्वर्ट हो जाती है हैप्लॉयड सेल्स, हैप्लॉयड सेल प्रोड्यूस करती है. दैट इज द मेन फंक्शन ऑफ द meiosis. पूरी बॉडी में वो हमें नंबर ऑफ क्रोमोसोम एंड अमाउंट ऑफ डीएनए दोनों को वो डिप्लॉयड स्टेज से हैप्लॉयड में कन्वर्ट करके देता है और उसी से गैमीट्स भी बनते हैं, meiosis से गैमीट बनते हैं. ठीक है, सो द होल प्रोसेस of the meiosis that occurs during a specific duration of time. It occurs at specific duration of time. स्पेसिफिक ड्यूरेशन ऑफ टाइम, यानी मियोसिस जो है वो एक पर्टिकुलर टाइम पे होती है, वो थ्रू आउट द लाइफ नहीं होता. रिमेंबर दिस पॉइंट. ओके, नाउ सी इफ वी टॉक अबाउट फर्दर द मियोसिस प्रोसेस, if we talk about the meiosis, just a moment. So if I tell you the key features of the meiosis, okay, the key features of the meiosis are as follows: that meiosis involves the two sequential cycles of nuclear and cell division called the meiosis 1 and meiosis 2, but only a single cell cycle of the DNA replication. It means during the whole process of the meiosis, तो इस होल प्रोसेस ऑफ मियोसिस में क्या होता है, इसमें टू सीक्वेंशियल साइकिल्स हो जाते हैं, यानी टू सीक्वेंशियली ट्वाइस जो सेल है वो डिवाइड होती है, जिसमें न्यूक्लियर डिवीजन होता है पहले और उसके बाद जो है साइटोकाइनेसिस हो जाती है 2 बार, और उसके बाद ये दोनों टाइम्स जो है 2 फेजेस में चलता है थ्रू आउट द मियोसिस. सो दैट इज मियोसिस 1 एंड मियोसिस 2. मियोसिस 1 एंड मियोसिस 2. तो मियोसिस 1 और मियोसिस 2 में 2 फेजेस में ये मियोसिस कंप्लीट हो जाता है. ठीक है. नाउ इफ यू गो टू द होल प्रोसेस ऑफ द मियोसिस, यानी 2 cycles of meiosis, meiosis 1 and meiosis 2, in both these processes of meiosis, only in one meiosis, that is the meiosis 1, ठीक है, during the meiosis 1 this DNA replication occurs. DNA replication occur in the meiosis 1 of the phase, interphase. Then after that in the meiosis 2 the DNA replication doesn't occur. So that is why this DNA replication occurs once during the process of the whole meiosis. Remember this point कि पूरी मियोसिस के दोनों फेजेस में, मियोसिस 1, मियोसिस 2, इसमें DNA रेप्लिकेशन जो होता है वो एक ही बार होता है, वो होता है फर्स्ट मियोसिस में. रिमेंबर दिस पॉइंट. So let me take you through the first meiosis 1, the first phase of the meiosis, that is the meiosis 1. If we talk about the meiosis 1 beta, so meiosis 1 में meiosis 1 जो है इट्स अ टाइप ऑफ डिवीजन जिसको हम बोलते हैं इट्स अ हेटेरोटाइपिक डिवीजन. क्या होता है? हेटेरोटाइपिक डिवीजन. इसमें हेटेरोटाइपिक डिवीजन इसलिए बोलते हैं, इसमें न्यूक्लियर डिवीजन अलग होता है, साइटोकाइनेसिस डिवीजन होता है, क्रोमोसोमल डिवीजन डिफरेंट तरीके से और डिफरेंट टाइप्स ऑफ क्रोमोसोम इसमें बन जाते हैं जो सेम नहीं होते हैं. सो इट इज कॉल्ड द हेटेरोटाइपिक डिवीजन. ओके, बट if we see the meiosis 1, meiosis 2 को बोलते हैं homotypic division. It's called heterotypic, that's called homotypic because उसमें nuclear division, cytokinesis division और जो chromosomes होते हैं, number of chromosomes same रहता है, chromosomic structural जो है वो ज़्यादा change नहीं होता है, वो equally रहता है जैसे mitosis में रहता है, तो उसको बोलते हैं homotypic division. So meiosis 1 को बोलते हैं heterotypic division. So this is the heterotypic division और इसको दूसरा नाम भी बोलते हैं, it is called reduction division. Meiosis 1 is known as reductional division because this is the phase, meiosis 1 is the phase जिसमें रिडक्शन हो जाता है, यानी नंबर ऑफ क्रोमोसोम्स रिड्यूस हो जाते हैं डिप्लॉयड टू हैप्लॉयड, डिप्लॉयड टू हैप्लॉयड. इसीलिए इस फेज को रिडक्शनल डिवीजन फेज भी बोलते हैं. रिमेंबर दिस. नाउ meiosis 1 जो है it leads to the reduction in the chromosome numbers. The chromosome number becomes reduced. Division of chromosome does not occurs in the meiosis 1 and only segregation of homologous chromosomes takes place. That means it says that during the process of the meiosis 1, the division of chromosome does not occur. It means the chromosome, if this is a chromosome structure, like you know that chromosome structure जो है, you know that about the chromosome structure. क्रोमोसोम ऐसा होता है, ठीक है ना, जिसमें दो क्रोमेटिड्स होते हैं. So it means the division of chromosome does not occur. It means the division of chromosome like this, the chromosome is not going to divide during the meiosis 1, and only segregation of homologous chromosome takes place. So during the meiosis 1, two chromosomes which are morphologically similar, one is the maternal chromosome, one is the paternal chromosome. These two chromosomes, chromosome 1, chromosome 2, they are morphologically similar. They come closer during the process of the meiosis 1 and led the formation of a pair. That pair is known as homologous pair. It is known as homologous pair of chromosomes. So during the meiosis 1, this homolog pair of the chromosome, they get segregate, like this chromosome goes this side, this goes this side, but the chromosome itself is not divided. Chromosome itself is not divided. Remember this point, the centromere of the chromosome is not going to divide during the meiosis 1. But what happens exactly? The chromosome goes separate. This chromosome and this chromosome, they get separated. That is called the segregation of homologous chromosomes. Remember this. Now if you talk about meiosis 1, the meiosis 1 is initiated after the parental chromatids have replicated to produce identical sister chromatids at the S phase. Now let me take you through one thing. I will give you the explanation of on the whiteboard that how exactly what changes occurs during the meiosis 1 beta. Okay? So let me off this PPT section. I will just minimize it and I will take you through the real board presentation now, because at background, if this content will be at the background, you will not be able to see properly the diagram which I am going to show you. Okay, so let me minimize it. I'll use the whiteboard here. Fine. See, now if I go with like, so let me take you through the different stages of meiosis 1. Meiosis 1. Okay. So meiosis 1, sorry, there is the whole meiosis get completed in different phases, different stages. So in the similar manner, the meiosis 1 is the largest phase of the meiosis, and it gets completed into by different phases. It gets completed into different phases. First phase, there is a phase called prophase 1. Prophase 1. And in the same manner, after that, there is a metaphase 1. Metaphase 1. Then there is the anaphase 1. Third phase. And there is a telophase 1. Telophase 1. These are the 4 stages in which the whole meiosis is going to be completed. Remember this. Now, suppose this is the cell. This is the cell, diploid cell. This is a diploid cell, and this diploid cell is now undergoing to the meiosis. This will enter into the meiosis. So it will enter into, it has to enter into the meiosis 1. Sorry. So this cell which has to enter into the meiosis stage, this cell is known as meiocyte. It is known as meiocyte, and this cell is a diploid cell. Suppose this is a germinal cell, okay, and this germinal cell, it has to enter into the meiosis phase now, meiotic phase. So that cell will be called as a meiocyte cell. Inside that cell, there are the two sets of the chromosomes, one paternal, one maternal. So it's a diploid cell. It's a diploid cell. But before entering into the meiosis 1, this cell, meiocyte cell, it has to complete the interphase. It has to complete the interphase stage. So the same cell which will enter into the meiosis, it has to enter first into the interphase stage. So this cell has to complete the interphase stage. Now interphase stage, I have already taught you the interphase stage, beta. So interphase stage is a preparatory phase. It is the preparatory phase. It's a preparatory phase, okay, you know that. And this interphase stage, it gets accomplished by 3 phases. That you also know that it gets completed by 3 phases: G1 phase, S phase, and G2 phase. That you already know, you have gone through this concept, what happens during the interphase, okay. So the same cell there in the mitosis which was entering the interphase, in the same manner this meiocyte will enter into the interphase and will complete its G1 stage, its S stage, and its G2 stage. G2 phase, okay. So in the same manner, in the G1 phase, what changes occurs there? You know that protein synthesis occurs, okay, lot of nucleotides are going to be prepared, major organelles are going to be developed in this system, okay. Mainly when they enter into the, then there are different checkpoints, you know that. Then cell enters into the S phase, synthetic phase. In this synthetic phase, here the DNA replication occurs. The DNA replication occurs. So you know that if this, if I give you the example of human cell which enters into the meiocyte, and if that human cell has diploid number of chromosome, so this meiocyte will be having 46 number of chromosomes. 46 number of chromosome, because it's a diploid cell, okay beta. So this diploid cell, when it enters into the G1 phase, then in the G1 phase, you know that number of chromosome remains the same. Number of chromosome will remain the same. So there is no DNA replication occur. So amount of DNA will be also considered as the, you know that 2x, fine, diploid. Then the same, it enters into the S phase, and in the S phase, then DNA replication occurs. So DNA amount goes doubled here. DNA amount goes double. So if there were a 2x amount of a DNA in the G1 phase, then it becomes 4x amount of DNA in the S phase. In the S phase. So in the S phase, you know that already I have taught you in the interphase, the amount of DNA goes doubled here, the number of chromatids gets increased here, but the number of chromosome will remain the same. There is no change in the number of chromosome. Remember this point. And the same situation will remain in the G2 phase. When the G2 phase gets started, so inside the cell, there will be the same number of chromosome, 46 number of chromosomes. It will be same there. But amount of DNA has been increased. So it means double-stranded DNA molecules. It will be there. How much? It will be there 92. That will be 92. So it means each chromosome has the 2 chromatids. Each chromosome has the 2 chromatids. So it will say number of double-stranded DNA as well as amount of DNA. Amount of DNA. So the amount of DNA, it is already 4x in the S phase. So it will remain as the same 4x. It will remain at the same 4x. So this is the change which occurs during the interphase of meiosis 1. Before the meiosis 1, this phase is called the interphase stage of the meiosis. So these changes occur inside the cells in the G1 phase, S phase and G2 phase. Now once the interphase is completed, once the interphase is complete, तो जब ये interphase complete हो जाता है तब ये myocyte cell जो है, now this cell is ready for the cell division, or it will enter into the meiosis stage. Meiosis 1 will get started. Okay, so this meiosis 1, now when the enter myocyte, this myocyte cell, diploid cell will enter into the meiosis 1. So the meiosis 1 gets completed in different phases: prophase, metaphase, anaphase, telophase 1. Okay, so what changes occurs in the prophase 1? So let me talk about the meiosis 1 का first phase हम discuss करते हैं, that is the prophase 1. That is the prophase 1. Okay. So let's talk about the prophase 1 of the meiosis 1. Now prophase 1, it is the largest phase. This phase, it is considered the largest phase of meiosis. Largest phase of meiosis. So this phase takes some more time. Okay, and this phase gets accomplished into the different stages. Fine. So during the prophase, it gets completed into different stages. By different stages, there is a first stage, and it is called leptotene stage. Leptotene stage. Okay. Then there is a second stage. It's called zygotene stage. Zygotene stage. Then there is a third stage. It's called PACEKashmir stage. PACEKashmir stage. Okay. Then there is a fourth stage, diplotene stage. It's called the diplotene stage. Then there is a fifth phase or fifth stage. This is called diakinesis. This is known as the diakinesis. Okay, so these are the five stages of the prophase 1. So it means the prophase 1 gets completed in these 5 stages. Now what happens in these 5 stages? What changes occur within the cell that you need to remember? Okay, so let me take you through the first leptotene stage. Okay, leptotene stage is the stage where you see that if this is the cell, myocyte cell, which enters into the meiosis of the prophase 1 of the leptotene stage. So this cell in the leptotene stage, this cell, it has the plasma membrane outside. Okay, it has the plasma membrane, and in the center there is a nucleus. Suppose this is the covering of the nucleus. This is the covering of the nucleus, which is called nuclear envelope. Okay, within the nucleus there is a small nucleoli like structure going observed within the nucleus, and it is called nucleolus. That you know that already, nucleolus. Both nuclear envelope and nucleolus, they are going to appear. They appears during the early leptotene stage of the prophase 1. They remains intact. They are not going to disappear. Now within this nuclear envelope, inside the nucleus, the cell contains a genetic material. Okay, so inside this nuclear envelope there is a genetic material in the form of the DNA, and that DNA is going to be condensed form like this, and different DNA strands are going to be formed in the shape of the chromosomes. The shape of chromosomes, okay, like this. So the chromosomes which are going to be observed here, these chromosomes are thread-like in the structures. They are these chromosomes in this stage, they are thread-like. They are thread-like, but they are thick, condensed. They are the condensed, thick condensed form, okay. So the more and more condensation of this chromatin material occurs inside the nucleus with the help of some special proteins, okay, like you have already gone through the histone proteins, fine. These proteins, with the help of them, they lead the more and more condensation of the chromatin material. Then these chromatins, they get aligned, gets attached with the centromere, led the formation of the chromatids. तो वो chromosomes के chromatid बनाते हैं. But in this stage, lactotin stage, what occurs that outside of the nuclear envelope, there is the presence of centrioles, two pairs of the centrioles, which also starts to divide. They also starts to divide. So the two pairs of the centrioles are moving towards the opposite pole of the cell. Opposite pole of the cell, they have started formation of the esterase. Now towards the centriole of the cell, towards the anterior centriole of the cell, where these centrioles are located, towards that area, most of the endings of these chromatids, they move towards that centriole. They move towards that centriole. They show the orientation. They show the orientation towards that centrioles. Then in that case, if you see these chromosomes inside the nucleus, they look like this, that most of the chromosomes, they're one of the end of these chromosomes, it move towards the centrioles, towards. So in that way, when the one ending go towards the centriole and other endings of the chromosome remain scattered inside the nucleus, then it gives the appearance of these condensed chromosomes. The chromosomes here in this phase, lactotin stage, they appears like a ball of वूलन बॉल, जैसे वूल की एक बॉल होती है वैसे लगती है, बट इसके एंडिंग्स जो होते हैं, देयर एंडिंग्स आर टुवर्ड्स द सेंट्रियोल्स, उनकी एंडिंग्स टुवर्ड्स द सेंट्रियोल होती है, एंड इट appears like flower bouquet, So ये स्टेज में जो क्रोमोसोम्स अंदर होते हैं वो एक फ्लावर बुके की तरह दिखते हैं, ठीक है, बिकॉज उनके सारे क्रोमेटिड्स के जो एंडिंग्स होती है वो टुवर्ड्स द वन साइड जाती है, टुवर्ड्स द सेंट्रियोल, बाकी थोड़ा सा ऐसे इलेबोरेट हो जाती है डिफरेंट डायरेक्शन में. सो इट गिव्स द अपीयरेंस ऑफ द फ्लावर बुके. इसीलिए इस फेज को जो है इट इज समटाइम्स आल्सो नोन एज फ्लावर बुके फेज और फ्लावर बुके स्टेज. It is also known as the flower bouquet stage. ठीक है. So these are the changes occurring in this lactotein stage. Okay, changes are like these: the nuclear envelope, nucleolus remains intact, chromosomes becomes more and more condensed, thick, okay, and it gives the appearance of the flower bouquet. These are the changes which you have to remember about the lactotein stage. Okay,